Welcome to The Explainer. Today we are digging into a modern-day scientific detective story. And the crime scene? Well, it's a very public place. A GitHub issue thread. It's this incredible story about a single software bug that was holding up critical cancer research and how a community solved it right out in the open.
So think about this for a second. How on earth would you find a typo the size of an entire chapter in a book that has 3 billion letters? It sounds completely impossible, right? But that is the monumental challenge that cancer genomics researchers are up against every single day.
Now, that book with 3 billion letters is, of course, the human genome. And the typos that these researchers are looking for, well, they're not just small spelling mistakes. We're talking about massive errors called structural variants.
Yeah, these aren't just single-letter mix-ups. This is like a whole chapter of your DNA getting deleted or copied and pasted in the wrong spot. These are huge, huge rearrangements of your genetic code that can have catastrophic consequences, and they're often what drives diseases like cancer. So finding them, absolutely critical.
To find these things, scientists have to use some seriously powerful software. And one of the real workhorses, one of the key tools in this hunt, is a program called Delly. You can think of it like a specialized digital detective, designed to scan just enormous genetic data files. We're talking terabytes of information, to pinpoint the exact location of these disruptive structural variants.
But what happens when the tool itself, the very thing you're relying on, becomes the problem? This brings us to a public call for help, basically a digital message in a bottle, posted on August 8, 2018.
You've got this researcher who goes by the name F.P. Barthel, and they are just completely stuck. A critical analysis that should really only take a few hours is stretching into days and then failing after 72 hours. It's threatening to derail their whole project, and that's when the tool's own developer Tobias Rausch steps in to investigate.
Okay, so the investigation begins. And like any good detective, the developer starts with the usual suspects. His first move is to question the evidence, the quality of the data itself. He's looking for the common culprits that might be slowing things down.
He first suspects a problem with how the DNA was actually prepared for sequencing. You know, it's a super common issue. It's kind of like the tech support question, did you check if it's plugged in? But nope, that wasn't it. A dead end.
The researcher checks and reports back that the insert sizes look totally normal, so the developer has to dig a little deeper. He asks a much more specific and, as it turns out, a very revealing question. And this is the question that cracks the entire case. It zeroes in on what seems like a tiny, super technical detail, but as we're about to see, it has massive, massive performance implications.
So with that definition, you can probably see where this investigation is going. The problem wasn't in the data at all. It was in how the tool was interpreting that data, all because of a flawed assumption about these so-called batch numbers.
And boom, that's the aha moment. With that one question, the developer realizes the tool was basically living in the past. It was designed with an old-school assumption about how DNA sequencing was done, an assumption that just isn't true anymore.
See, back in the day, you had to run a sample multiple times and then pool all that data just to get enough to work with. This meant a single file could have dozens of these read groups or batch numbers. Delly was painstakingly built to track every single one. But today, the technology is so much more powerful, you only need one run. So the tool was wasting all its time, all this computational power, searching for ghosts of sequencing past. So what's the fix? Well, it's actually pretty simple. The developer just updated the tool. He changed the default setting to match the reality of modern science, to just assume one library per sample. And that simple switch instantly fixed the crippling performance issue.
And just like that, case closed. The researcher is unstuck and can get back to their incredibly important work. And because this all happened out in the open, DLI is now faster and more efficient for the entire global scientific community. It's a win-win.
Okay, so the bug is fixed. But why does this one little interaction on GitHub really matter in the grand scheme of things? Well, to understand that, we need to look at what this tool actually produces. It all comes down to this one output file. It's called a variant call format file, or VCF, and you should think of it as the treasure map. This file is the final report that points scientists directly to what makes a cancer cell genetically different from a healthy one. Okay, so at first glance, this looks super technical, right? But just think of it as a set of precise coordinates on our treasure map. Each line tells a scientist exactly which chromosome to look at, the exact position on that chromosome, and what kind of genetic error they found. So by reading this file,
Researchers can finally see what's really going on inside a tumor. They can see the exact chapters of the DNA book that have been deleted or duplicated or scrambled. They can pinpoint the specific structural variants that might be driving that disease. And this is why it all matters so much. This is the ultimate payoff. There is a direct line from a single programmer fixing a bug on GitHub to a doctor in a clinic being able to understand a patient's unique cancer. This is the kind of data that fuels personalized medicine, the development of therapies that are targeted at the specific genetic fingerprint of a tumor.
You know, this whole story is just such a great reminder that modern science isn't always happening in some sterile, isolated lab. It is a messy, collaborative, and incredibly powerful open-source effort. And a lot of the time, it's happening right out in the open for all of us to see. Makes you wonder, what else is being solved one GitHub issue at a time?